reduction today, so you have that one down too, right? Honestly, I promise that that's the way to getting a better grade. So really, like, if you didn't do that, you really want to make sure that by this weekend you know that. So we're heading into bacteria, and as I was mentioning, there's going to be, like, it's somewhere, don't hold me to 45, but it's somewhere around 45. It may be between 45 and 50, where you will have to know the names of the organisms and what they're associated with, and maybe if there's specific characteristics. I told you guys this last time. That's why I said learn that other stuff, because you have enough stuff to do. And really, this particular exam will get people every time, but it's not really that hard if you keep up with it. So all this other time, I think, like, you probably don't keep up with it like you know that you should, and then after that weekend when you're studying really hard, you wish that you would have started studying sooner. If you try to memorize 45 things, or about 45 things to 50, plus the stuff that we did last time, in the weekend before the exam, you will fail the exam. This is always the past, I would say, couple of years, this has the lowest scores of any exam. And every single person will say that the reason why they didn't do as well as they think they should have is because they didn't start learning the names of the organisms all along. So if we have a lecture, sometimes in one lecture, there'll only be like 10 names. You can go home and learn 10 names, or 8 names, or 5 names. If you divide it up and you learn it like that, and you do it in small sections like that, then the test will be an easy test for you. Like, it'll be easy. What's hard is trying to learn too many names at one time. You will not have to write out the names. You will just have to be able to match the names with whatever their disease is or whatever their characteristics are. This is our chance to really talk about individual organisms that are representative of the diversity in bacteria. So we kind of weigh it a little bit heavier on disease-causing organisms, just because some of them will already be familiar to you. Some of them you should get out of an introductory microbiology class, knowing a little bit about botulism, or salmonella poisoning, or some of the ones like listeria, which, like, every other week they've been recalling things. There was another recall for listeria today. So you should know something about those kinds of diseases. But some of them will also just be about different groups of organisms, because that particular group is a representation of the diversity that we see in bacteria. And that's something that that I've been saying kind of over and over and over about, oh, there's so much diversity, you can never say all, that kind of thing. So that's what you kind of are going to be leading into. So you want to make sure that you take really good notes, because if you started printing out these notes, you'll know that there were a lot of notes in some of the categories. And as we go through, I'm going to be saying frequently that I've left these different examples in the notes because I want you to know that they exist, but I'm not going to ask you to memorize them. So when you're talking about at least 45 examples, basically what happens is every day somebody comes in, so what are the organisms that we're supposed to know so far? And I'm not going to just be able to say, like, right off the top of my head, all 45 of them, and that you don't have to memorize this or that. So please, like, do whatever you need to do to make sure that you're staying up with those. Also, keep in mind that if I talk a lot about a certain example, like, maybe I'm not going to be able to test you, like I'm saying I want you to know all 45 or 50, but maybe there's going to be more information on certain ones, or there might be a couple of questions on certain ones. You're going to be noting that if we spent a lot of time talking about a particular example, then you know that's going to be on the test. So then you know that you have to memorize that one, and that you have to prioritize. So I know some of you are getting the hang of that, but I mean, I'm just gonna say it one more time, because there are certain things that it's just like I'm handing it to you. You know, I told you guys about that viable but not culturable. So you guys like should make sure that when I'm talking about an example, and this is the time where a lot of other people will participate too, where they feel like, oh, well, I had, you know, a relative that had this and this is what happened, and, you know, or this is going on in the news. So sometimes we'll spend more time talking about a particular example in this particular semester than maybe I would have at another time, because it's just relevant at this particular time. So that's kind of what this next section is all about. It's all about that diversity. And now, again, you've heard me say diversity, diversity, diversity. Now again, I'm saying it again. Could there be an essay question on the exam about diversity? I've done that before. Because if there's anything that I want you to be aware of, is the fact that there's a lot of diversity in the area of bacteria. So you're thinking of the fact that, yes, we're talking a lot about disease-causing organisms, but there are all these other really unique, interesting organisms already. When we talked about the bacterial cell, you see that kind of diversity, that there's organisms that don't have a cell wall. There's organisms that have certain other structures. There's organisms that live in all these different ranges, right? Like you had to learn that last time, you know, that there are ones that live, you know, under high pressure conditions, and ones that live in boiling acid and, you know, such. That's the kind of diversity that you see, but this is the time that we can actually talk about where they're classified and, you know, what maybe some of their names are. So sometimes there'll be a group, and sometimes it will be the actual genus and species. But this, I think, is the fun part, because we do get to talk about examples. So, like, hopefully you'll look at it that way, but I'm just kind of giving you, like, this talk on day one, because I know that everybody will be like, oh, dang, I know you warned us about that, oh, you know, and it'll be, like, not just struggling students, it'll be good students, it'll be everybody that just, like, lets themselves get behind. It's the key is just learning a few at a time as you go. So this chapter 17, that's just finishing up where we talked about classification and taxonomy and such already at the beginning of the semester. This is just the end portion of that chapter, just giving us a chance to kind of remind you of what goes into classification. You are right, taking this class at a time where we're actually in between classification, just like we were when it was the eukaryotic organisms, and we're switching over to that International Society of Protostologists. We are now in Bergey's classification, the second edition, but we still are using a lot of stuff from the first edition. So like even in other classes, the students might be memorizing the first edition classification, which I don't have you do because it's definitely being changed. There's 5 books in the second edition, and only about 3 of the books, I think, right now have been published. I mean, the information is out there. A lot of organisms have been reclassified. They basically really just took everything and looked at every organism, so it just has taken years and years. So this is already something that's been going on for a good, I don't know how many years, what does it say? The first edition was in 1984. The second edition, it doesn't say on there, but it's still in the process of being really, like, with every organism evaluated and possibly reclassified. The same things happen where they went from a classification that was really based on characteristics. So if you had something that was gram-positive and it was a spore former and it, you know, was anaerobic and such, you were likely to find it in that particular classification. The way that things were classified before too is that many of the classifications were things like gram-negative non-spore forming rods and cocci. That was the name of the classification. So it made it very easy to use because really it was very user-friendly. A lot of the characteristics of the organism were already named in the classification. So it's not like that at all anymore. Everything now is based on what you think. Usually the genetics of the organism. So things change dramatically. But they change in a way that it doesn't make it as usable for one thing, for clinicians, people who are working with people who have the infections, because it's much easier to think about an organism that is a gram-negative rod, you know, because that would be something that would already maybe say something about, okay, well, it's gram-negative, so it has this kind of cell wall, so it's going to respond a little better to this particular type of antibiotic, and we know that these particular organisms produce these types of toxins, and so that tells us something. We have now a category in which you have something like organisms that don't have a cell wall, the mycoplasmas, that are now classified in a same grouping basically with endospore forming organisms. So you have like some of the most resistant organisms being classified with ones that would be much more susceptible. So it will be a long time to where people are actually really utilizing the newer classification, even though, again, it's already been going on for years. So going back to this beginning, this is really just showing a little bit of the overview of what has happened. And again, I've mentioned how we started off with that, you know, first cell, the rod-shaped, anaerobic, thermophilic organism, and now we have all this diversity. How did that occur? We talked about some of the methods last time, and that's really what this is about. Bergey's Manual of Systematic Bacteriology, this is basically what we use. And I think, well, since you didn't have the library orientation, you didn't really get to see a copy of Berkey's manual, but you probably have come across that or maybe heard somebody mention that. That's where you can really look up everything. So you see this detailed work containing descriptions of all bacterial species currently identified. You can actually look up in Berkey's manual some of the results of the tests that you guys have been doing. And you might think, well, why did we do all those tests if we could have just looked them up? But there's a reason for that. There is so much diversity, even in those species and strains of organisms, that when you look up things in Berkey's manual, and I've seen some of you guys look up things, so some of you have probably come across the same situation. It may or may not ferment glucose. It may or may not. Some species, some strains, you know, so you end up like even more confused because you look it up and you realize that there are certain physiological tests that are really representative of that particular grouping of organisms, and then sometimes they are just a range. Like it just varies between the particular organism that you are working with. So basically in our lab, we test the organisms that you are working with. So at least you're more likely to have the most accurate things. You don't want to be looking up and finding out, oh, it may or may not do that, it may or may not do that, it may or may not do that, because that's kind of what you find out. If we really had a particular strain in which we purchased from something like the ATCC, the American Type Culture Collection, American Type Culture Collection. They are the ones that maintain samples of all the known bacteria, the American Type Culture Collection. For a lot more money, we can purchase an exact strain of a particular organism that at least it's going to be much more likely to be reliably responding to these different tests. So sometimes when we're working with like E. coli, we're getting E. coli from a biological supply company, then we start maintaining our own, and it's just sort of the run-of-the-mill ordinary E. coli. Or we could pay 3 times the amount and we could get like E. coli type strain B. Or we could pay, 20 times the amount and we could get that specific one in which we know it responds to particular tests a certain way, because there still is a lot of genetic diversity between one species and another organism in the same species. So one organism, another organism in the same species. So with bacteria, they can be identified down to strains, and some of that is at kind of the end of these particular slides. So the first edition, it's really, let's see, it's currently in its 9th edition. That's just because there's updates in here, but the first edition is where they came out, and that had all the classifications. Yes. American Type Culture Collection, and it's just abbreviated as ATCC. The second edition you see here, largely phylogenetic rather than phonetic, right? So it's more that it's really relying on all of those biochemical and genetic tests to determine what classification it should be in. And in this one too, the prokaryotic cells are really in archaea, completely separate. It used to be that the archaea was just one particular division of the first edition. Now there are 2 domains, so the archaea are in there, but they have their own book. So if you started looking at this in your textbook, then you know like chapter 18 is a whole chapter on the archaea. So instead of them just being like one division in the first edition, now they're the whole book, and they're really treated separately because they are in another domain. So what I want you to know from this is just what is the difference between the first edition and second edition, which is really basically the same kind of information as what you had to write your essay on. So how likely would it be that I would ask you to write an essay about that? Not likely, because I already made you write an essay about that. But I could ask you a question about it, because I just want you to be aware of that. And to be aware of the fact that we are in a process of changing. So therefore, if you are looking something up, you're gonna see that kind of information where something was classified a certain way, and then why are they saying it's in this? When I'm reading this, it says it's in something else. That's why. Because there's really a big lag time. I mean, honestly, it was just like 2 textbooks ago where we got our new textbook, and you open up the textbook and it's like, what? All of a sudden we have, a completely different classification. So all of these next slides are really just talking about how the classification is done. So you can read through those. I'm not going to specifically talk about each thing, but you can see this looks familiar to what we went through before. Molecular characteristics. The nucleic acid-base composition. This G plus C, this is worth noting that this is important, and there are two books, the high G plus C and the low G plus C. And that's because these particular numbers are at a much bigger range than what you see with eukaryotic organisms. So the G plus C, the percent G plus C content, that is something that is really used widely in with the bacteria. And you'll see that because there's 2 of the 5 books are based on the G plus C content, percent G plus C. So there's a range much broader among bacteria. But you still see in here variation within a genus less than 10%, right? So a genus, that means like if you had, Bacillus cereus and Bacillus subtilis. You can see they have similarities, but they also have differences. But their G plus C content, because they're in a genus, is usually still like more like 90%. Looking at the ribosomal RNA. And now looking at this, okay, we know about the domains. We know that we have bacteria and archaea. In the domain, they have the same, levels: phylum, class, order, family, genus, and species. So normally we really are talking about genus and species. Sometimes there are strains or subspecies. You will see that everything is organized according to the phylum. Some things, it's frequently, you'll see class and families. Really, for the most part, I think I can say that I am not going to ask you to know phylum. It's even probably, maybe once in a while it might come up with family, but I'm not going to ask you to know the phylum. You wait till you see all of these terminologies that go along with it. There's only so much I can ask you to know. I'm not going to ask you to know the families, but you'll see that everything is organized according to families, and I've stuck to the order in which it's presented to you in your textbook. But I'm not going to ask you the families. It's just too much. So by the time that we get through talking about all of this, I'm going to present it to you in the way that it's presented in the book, according to the books of Bergey's second edition. But for the most part, I'm either going to ask you to know about a particular group or a particular example, rather than exactly what its classification is. Okay, so keep that also in mind. There's no way I can ask you any more than that. I'd love for everybody to memorize everything about it, but there's no way I possibly can, so I'm not going to ask you that. And this just kind of gives you an example. The domain is bacteria, but you see this. There's phylum, the proteobacteria. But we're going to be all the way down. I will be asking you about Shigella, Yersinia. You already know about Serratia, Salmonella. We working with Proteus, Klebsiella, Escherichia, and Enterobacter. And then when you go further, you get the genus and species. Okay, I'm not going to be asking you that they're part of Proteobacteria, and that they're part of beta Proteobacteria, and that they're in the Enterobacterales, and that they're in the Enterobacteriaceae. Though you will come across this pretty frequently, because this particular family is kind of spoken about frequently. But you see all those levels? I can't ask you all of that. So sometimes we'll be talking about a group, which doesn't even really fall exactly into here. But most of the time we're going to be talking about the genus and species. And you know what a genus is, you know what the species is. But here's a chance for me to kind of show you that you're looking sometimes at 70% homologous in the same species. And when you see that, 70%, that means that there's quite a bit of diversity in the species. That's why I could say that when we're talking about, run-of-the-mill E. coli versus E. coli B versus any of the other ones, there's a lot of diversity in that species, which you don't see in eukaryotic organisms because you're not getting all these subspecies. But it does help you to understand what happens in your lab when you're working with a particular organism and you're like, well, we both inoculated something with this particular organism. Why did they get this result and I didn't get this result? You already know to think about, well, maybe you didn't inoculate it properly, maybe the media was old, maybe it was contaminated. You have all these things, but you also have the fact that sometimes it just doesn't. Sometimes you just don't get the reaction you should because there still is a lot of diversity in that. Plus, on top of it, added into it, you have the knowledge now about the growth curve. So you know when you're using these old cultures that it's not really far-fetched to believe that they could have mutations and be different than the organisms that were first put in there. So it's kind of like 3 different things all coming together that help you kind of understand what's going on. and maybe understand even more about what happens when people get infections and the physicians have a hard time treating them. We tried this and it didn't help. We tried that and it didn't help. Now we're using this combination of these two things and we're trying this other thing. Because some person can have an infection, it's the same organism, but there might be things that are different about that organism. And on top of it, with humans or anybody that has the infection, you have the immune system and you have a lot of other things too, the difficulty in treating it, the antibiotic resistance and such. So it's like you have to understand all of this to really get the big picture. So I haven't lost anybody on this, right? You understand what I'm getting at. So strains, this is kind of pointing out that you can have these different strains and they could be different morphologies, different antigenic properties, different biochemical and physiological properties. And these are ones that are known. When you say antigenic properties, you know that they would then stimulate different antibody productions in somebody. So if you were exposed to a strain of something, even though you had been exposed to that organism before or had that infection before, you can have a different strain. It has different antigenic properties. It doesn't elicit the antibody response from that exact antibody. So it's like starting over again. The type strain is kind of what I was mentioning about the American type culture collection. They're the ones that keep, basically it's like a library of all of those particular identified strains. And this is just a slide kind of showing about that percent homology. So we said in the same genus you might have 70%. These are just different examples from it's the same genus. And this is just different specific examples. So you can see already in this one, you went from 35 to 78%. Okay, so that's kind of like an overview, just so that you kind of know what you're expecting for going in here. So our first one are the archaea. You guys have your archaea notes. Okay, so with the archaea, this one is going to be fairly easy for you because you already know something about the archaea. On the last exam, you had to give some differences between the archaeal cell and the bacterial cell. So you know that structurally they are different. Also, we have some nice categories in here so that it's very easy for me to present this to you guys as far as the types of archaea. But when you go through and you look at this particular overview, they talk about the things highly diverse with respect to morphology, physiology, reproduction, and ecology. Best known for growth in anaerobic, hypersaline, pH extremes, and high temperature habitats. So we had said before, when we talked about the archaeal cell, that they did tend to be the ones that were more extreme. There were square bacteria, there were bacteria that were sheathed, or bacteria that enclosed in a slime layer that kept them together, or bacteria that were branched and such. And then you know that they lived in those extreme environments. You're going to pretty much see something like this in every book. Every time we start the new topic, they're going to say, oh, this is so highly diverse, because everyone is highly diverse. In fact, after we talk about the archaea and we talk about the Deinococcus next time, we're going to come across then Deinococcus, which is one of, if not the most extreme resistant organism. And that whole book is full of the same things. It's so highly diverse. So yes, the archaea are highly diverse, but again, you're going to see that in every category. You're not really going to come across any book in which they don't say this. So what you're going to have to know for the archaea are these 5 groups. So they're divided up into groups in here. The methanogens, and there's a slide at the end. Let's see. Not quite the end. Where is it? I must have passed it. You guys have it. It's probably sitting in front of you, right? A slide. The slide is this one. So number 12. These groups, the methanogens, halobacteria, thermoplasms, extreme thermophilic, sulfate metabolizers, and sulfate reducers. You need to memorize those 5 groups. So if we kind of go back to this, then you see this here. The methanogens, they just have archaeal in front of all of them, but methanogens, the sulfate reducers, the extreme halophiles, the cell wall-less, which are the thermoplasms, which that'll remind you over here, because even though that's thermoplasma, that's a particular genus, which you know because you can see that it's italicized, and the extreme thermophilic sulfate reducers or metabolizers. Okay, those are the 5 groups. I will expect you to know something about the methanogens, but I literally will have a blank and say, name the 5 groups of archaea. Okay, the only thing that I'll ask you to know something more about are the methanogens. These 5 groups, though, their titles tell you something about what kind of bacteria they are. You know, these all don't have cell walls. That's really the main characteristic of there. And if you read about the little characteristics in here, you can see like there's, you know, they're pleomorphic because they don't have a cell wall. But also they're thermoacidophilic, they're chemoorganotropic, facultatively anaerobic. All of those terms are things that you already had to learn about. So I can't ask you to memorize all of this about all the organisms. Again, I can't. There's way too much information. Okay, so I'm asking you to know just some of the representatives. But if you look through here, you can get some additional information. You can, again, understand. So that if I did ask a question about diversity, citing some examples, you would be able to bring up an example and understand that, yeah, wow, just in this one group, these are just the archaea. We haven't even started with the bacteria. Just in the archaea alone, there's all these organisms that represent the diversity. So memorize those particular names. These are the 5 major physiological and morphological groups of the archaea. When you see all this, here you can tell which things are the genus, you know, which things are the species, but these are giving more examples of the names of the organisms, which I'm not going to ask you. The characteristics you already had to learn about before in chapter 3. So the cell envelopes, flagella, lipids and membranes, metabolic things we, you know, kind of touched upon. You know how to determine what those particular words mean. But again, I'm not going to ask you to give me any more information about any of them, but all that information is here. Hyperthermophiles, they live by hydrothermal vents. They're acidophiles, they're dependent on sulfur. So you would see them at some place like Yellowstone National Park where you see these boiling acidic, sulfur springs. Those ones that are magnetic, remember the magnetosomes that were part of the cytoplasmic inclusions, the list. We mentioned that there were ones that was one of the inclusions. Here's an example of those. The cell wall is different. So there's really all that information that you've already been exposed to. Here's that particular slide. And now I just want you to know about the methanogens as an example. So you have to know those 5 groups, but I could ask you a question about the methanogens. And that's because really all the organisms that produce methane gas are in the archaea. So they produce methane gas. So you're going to make me carry those folks. You don't need to take them. So for those organisms that live in the intestines that produce methane gas, those are archaea. There's more and more evidence now that people that have certain digestive disorders probably have an overgrowth of methanogens, that they're now starting to link that to irritable bowel syndrome and other types of intestinal disorders. So this will be something that you'll hear a little bit more about as far as being associated with possible diseases, because it was just kind of thought that they were just responsible for causing people gas, but not anything more than that. Now they're really thinking there's something more than that. Also utilizing methane gas as a fuel source. So there's really a lot of research going into that, because we have all these animals that then are producing waste, all the humans producing waste. They have in many areas those methane gas recovery sewage systems and even individual toilets in some areas. So it's kind of like, well, we have this source, maybe we should start utilizing this source. So here, this slide 15 kind of gives a little bit more information about that important. And wastewater treatment can be used as a clean burning fuel and energy source. Also could be contributing to global warming. So it's just one of the more important of the archaea. It's kind of hard to say which thing is more important, because they all obviously live in a certain ecosystem where they are going to be beneficial. But this is just something because it affects people and it affects the environment, and it's something that we're doing a lot of research into. And so then as we go through, you can see there is some general information about each of these groups. So all you really need to know is what that name of the group is, and if there's a particular characteristic, like, okay, it's a thermoacidophile, it doesn't have a cell wall. The halobacteria, you know what halophiles are. The extreme thermophilic sulfate reducers, those are extremely thermophilic, right? And remember what thermophiles are. And this one also extremely thermophilic. And also again here too, you see that hydrothermal there. And that's all you need to know about the archaea. So any questions about the archaea? So now you know the overview about classification, have a little bit idea there, and now we can talk about some of the bacteria. Okay, so remember again, not asking you to know all these phyla and the names of the phyla. But as you go through here, you're noticing right away. I can't get a highlighter on here. For some reason, I got some weird communication that said that if I do something to this, it might cause harm to my computer. I don't know what's wrong with this particular one, but you can see here too, bacterial thermophiles already. So as I mentioned, you read that in Archaea and they make it seem like, oh, that's so unique to Archaea. But as soon as we start our next chapter, they're already talking about thermophiles. These grow in geothermal areas. And now, here we have the Deinococcine. So here's going to be your first organism to know in this group, Deinococcus. Okay, so I'd be highlighting that, but my highlighter won't work. Okay, Deinococcine. Because when you look at this, these are thermophiles too, but look at this, extraordinarily resistant to desiccation and radiation. This is something that's growing where that Chernobyl meltdown is, where nothing else is alive. It's able to survive extreme radiation. This is considered to be probably the most resistant organism known. You literally, like there's a whole scientific company that's devoted just to studying Deinococcus. You can go on YouTube and you can read like some of the talks that people have put out about, this is like even an organism that's like used in the whole debate of, you know, the evolution versus creationism. So there's a lot of information out there, but when you see this can survive 3 to 5 million rats, 100 rats is a lethal dose for humans. 3 to 5 million, and the fact that if they would be living in these extreme environments. Yes? No. They don't know why. They're trying to figure out why though. Because they think if you could figure out a lot of what's behind Deinococcus, that maybe we might have a lot of answers to what happened in the universe. And then when you look at this, isolated from ground meat, feces, air, fresh water, and other sources, but natural habitat is unknown. For one thing, in order to really do a lot of research, you have to like look everywhere, find organisms everywhere. There are billions of organisms. So if that's not an easy feat, that's why there's really a lot that's not known. There's a lot that are known about certain organisms, but there are a lot of information is not known because just the amount of time and money it would take to really study everything and look everywhere. So Deinococcus, when you look at something like this, I think people wonder, like, do I have to memorize all this, like produce acids from only a few? No, you don't have to know that. What do you have to know about Deinococcus? That it's really thought to be one of the most resistant organisms known that is able to survive these really extreme environments, and it seems to be found in a lot of different environments. I would hope that as I'm talking that you kind of picked up on those as being the facts that you would want to know. So as we go through, and they might say that something is anaerobic or something's microaerophilic or facultatively anaerobic, well, you have to know that about everything? You can't possibly. But when we talk about maybe bacteroides, because it's the most common anaerobe in our intestines and it's important from that room, then anaerobic would be an important characteristic. So that's again why it's kind of important to make sure that you are taking good notes, because I don't want you just trying to memorize like so much information that you don't need to know. Okay, I want you to be exposed to the information because it helps you kind of understand all of this, but you can't memorize everything about everything. There's just way too much. Now, the next ones that we're gonna talk about are gonna be the photosynthetic bacteria. So here we are first, like we started this. It doesn't really seem like, wow, photosynthetic bacteria, these used to be in a category all by themselves. Why? Oh, they were the photosynthetic bacteria. That made a lot of sense. And then we had the photosynthetic bacteria that produce oxygen, and then we have the photosynthetic bacteria that don't produce oxygen. So it was very easy to classify them all together. But now they're in this category which has this range of bacteria. That first it's the really resistant organism, now it's this, and then we're gonna go on to talk about chlamydia. That's also gonna be in here. Something that seems like it may not have a lot of relationship to this. But for this, I want you to know about two groups. Group one are the cyanobacteria. So you see here, the cyanobacteria, this is, a phylum, but it's also a genus. But I just want you to know about the cyanobacteria really basically as just a group. The cyanobacteria are known as the oxygenic phototropic organism. So if we go this, and I'm sure in your notes you probably can't read this particular chart or table, but here on the far right, oxygenic photosynthetic bacteria. Okay, and those are also called the cyanobacteria. Okay, and then I want you to know about the anoxygenic phototropic bacteria. So first, Deinococcus, that's a genus. Okay, now these are two groups. One is the cyanobacteria, the oxygenic phototropic bacteria, and the other one is the anoxygenic phototropic bacteria. Okay, so we had 5 groups from the archaea that included the methanogens, and now we have 3 things that you have to know. One is a genus, the other 2 are these groups. Is everybody clear on that?
No. Can you go over that one more time?
From the beginning?
No, just the last.
Okay. Yeah, because I'm going to talk about these. But one group is the cyanobacteria. Those are the oxygenic photosynthetic or phototropic bacteria. The other group are all these other ones here, which are the anoxygenic. These are the photosynthetic bacteria. The oxygenic ones produce oxygen. The anoxygenic do not. They produce other things. Okay, so these are important groups of organisms that live in aquatic environments. Cyanobacteria are the group of organisms that really are what you think of as algae. The cyanobacteria were always in the past referred to as blue-green algae. Then they started calling them blue-green bacteria. That's where cyanobacteria come from. Okay, so if you looked at a pond that looked like this, and you saw all that stuff there, you go like, oh, look at all that algae. When really in reality, the majority of algae that you see are cyanobacteria. There's still a lot of algae too, but a lot of the typical algae that you see are actually cyanobacteria. And they're really responsible for providing a lot of oxygen for our environment, a lot. But if you also went deeper into a pond, you'd find all kinds of bacteria. All the way, I really looked for a really good cross-section of a pond, and I could not find one that was related to this. But basically, at every level of a pond, you have bacteria that are using the wavelength of light that makes it to that spot in the pond. So the ones at the top use the wavelengths, they produce oxygen, and as you go deeper, they're using other wavelengths. Here where you have these that are then producing other compounds. So when you look at these, It shows the different photosynthetic pigments that they have. And cyanobacteria has chlorophyll A like all the other plants. So even though it's a prokaryotic cell, it has that similarity. And we may have mentioned it when we talked about the difficulties in classifying algae as an example in the maybe our very first lecture. And then when you go through here, you see we have the green sulfur, green non-sulfur, the purple sulfur, the purple non-sulfur, the aerobic anoxygenic phototrophs. So in this particular category then, what we're really looking at is photosynthetic bacteria divided up into two groups, the ones that produce oxygen and the ones that don't produce oxygen. They utilize different photosynthetic pigments, and they use different wavelengths of light, and they inhabit different layers of the water, so that literally at every layer of the water there are happily living bacteria there, utilizing that whole wavelength. You know how there's long going to short wavelengths. So the short, I think it's the shorter wavelengths make it down into the deeper area of the water. And see again, there's just so much information here. You can look at these. These are obligate photolithoautotrophs. I'm not going to make you memorize all of this. If you, in your 107 class, when you learn how to use a microscope, I'm pretty sure you looked at Anodina, because we always used to set Anodina out. That's this one here. This looks very similar to Volvox down here, but it isn't Volvox. Let's see if there's anything else on here that you have to know. No, that was it. Okay, so any questions about what you're responsible for knowing from that example? Yes.
I just want to verify the difference in this, the pigment, the layers of water, and
The wavelengths, yes. Everybody got that? Okay. Then our next example is going to be the chlamydias. So first, I want you to know about chlamydia as a group. Chlamydia used to be in its own classification by itself. It says phylum Chlamydia. It doesn't really matter. I'm kind of, again, like I talked about the cyanobacteria, I'm going to talk about chlamydia first as a group. Okay. Then I'm going to give you an example of chlamydia that you're going to have to know about. So I want you to know about chlamydia as a group because it is an obligate intracellular parasite. So the second thing down here, obligate intracellular parasite. Really, you will hear things about chlamydias and rickettsias, because chlamydias and rickettsias used to be classified together in their own category, but now rickettsias are in another book. And they were really discussed frequently because if these are obligate intracellular parasites, then why are we still saying that viruses are not considered living? Chlamydia cannot survive if they are not inside a cell. Okay? They actually cannot produce ATP. They have a certain developmental cycle, so if you were to actually look at them in different stages, they will look completely different. So here on this next slide, this developmental cycle, you have an elementary body and a reticulate body. There are just a lot of differences in chlamydia as a group. So that's why I want you to know that even though we've had a lot of changes in classification, chlamydias are always going to be chlamydias because they have a group of characteristics that are very unique. They're obligate intracellular parasites. They don't produce ATP, and they have this unique developmental cycle. So here on here's, in case you didn't see that, cannot synthesize ATP or NAD. And then I want you to know this organism as an example, Chlamydia trachomatis. Okay, so chlamydia as a group, and know the characteristics of the group, and then another thing is knowing Chlamydia trachomatis as an example organism. You can't get through this class without knowing something about chlamydia, right? You probably already know a good amount about chlamydia. You probably just didn't know that it has characteristics that make it, somewhat similar to a virus. Chlamydia really is, and you see here, you probably know it more as a sexually transmitted disease. Down here, the greatest single cause of blindness throughout the world. I don't have my notes with me to give you any statistics about this. I wish that I would have had them in there, but I'll let you know. But it is extremely common as a sexually transmitted disease. A lot of people suffer with chlamydia. There's an ugly picture of, what chlamydia can do if it gets in the eyes, and basically this is something that the mother can have chlamydia that can be passed to the baby. People can just pick up chlamydia just from, as you see here, transmitted hand-to-hand contact with infected soaps, and so on. It was difficult to culture chlamydia because it's an obligate intracellular parasite. So even if they did some kind of exam, like pap smear type exam, if you take a swabbing, the organism is then going to die off because the organism cannot live outside of a host. So it really wasn't until, I'm not sure what year it is, but I actually worked at the medical device manufacturer who developed the little brush that they use where they take a bunch of cells when they do something like a pap smear exam. It's, instead of it just being like a swab, it's like a brush. And basically what they do is it scrapes off a lot of cells. That way you have cells that have the organism in there. So culture tubes, I worked for a company that makes culture tubes, like when you get a throat culture or something. So they developed that, and that, like, I probably was there in, I guess it would be in the 80s. And it was like really just coming out at that time. So before that, they really couldn't see, except for looking at symptoms, whether somebody had a chlamydia infection. Plus, a lot of those types of bacterial infections kind of have similar qualities. So it's something that is kind of more new. That's why there's really a lot more information known about, the fact that it is something that is also sexually transmitted. As far as causing trachoma is then what causes the blindness. And this was something that I just remember in, when kids are in elementary school and they have to learn about the people immigrating here and going through Ellis Island and all of the different things, and how they took all the immigrants and lined them up and did all different kinds of, tests, because the U.S. didn't want to accept people coming to the United States that wouldn't be able to take care of themselves. And one of the things they had to do is have this little thing where they flip their eyelids back. Do you ever remember reading about that? And that was kind of, they used something that was kind of like a little knitting hook, and they lifted it back, and if they saw any redness in their eyelid, they were sent back on the boat. They weren't allowed to stay. That is this organism. That is trachoma. because if they had that infection, there wasn't any way to treat it, that person eventually was gonna go blind. There wouldn't be resources here to take care of somebody. So anybody who went through that process and they saw that, they were sent back. So there's just the ugly picture, like the book is